Hi, everyone. Thank you for giving me the opportunity to interview at UC San Diego. I'm very excited about interviewing for the research track, and I'm looking forward to sharing some of my work. Let me share my screen. Here we go. So I'm looking forward to sharing some of my work, which was done during my PhD at Johns Hopkins in the labs of Drew Pardoll and Kelly Smith. I was interested in looking at the role of regulatory T cells and how tumor-infiltrating Treg lineage is associated with response to anti-PD-1 checkpoint blockade and non-small cell lung cancer. So even though with the advent of immunotherapies, the trends in cancer death rates have dramatically reduced over the past few years, most markedly, we've seen this reduction in death rates in highly immunogenic tumors, such as lung tumors, as well as melanoma. Unfortunately, though, even with the improved treatment options and the overall trend in the reduction of cancer deaths, lung cancer in particular still remains one of the leading causes of cancer deaths, both in the United States and worldwide. And this is in part due to late diagnosis, but it's also in part due to a lack of response to the therapies that are currently available through various different immune mechanisms. So many groups have studied mechanisms for immunotherapy sensitivity. Immunotherapies unleash the endogenous immune system to have an effective anti-tumor T cell response, leading to tumor killing. In this study, I was particularly interested in the role of anti-PD-1 immunotherapies in non-small cell lung cancer. And specifically, I was interested in looking into the immune mechanisms of resistance to therapy, particularly the role of regulatory T cells and how they may contribute to enhanced suppression within the tumor microenvironments, overall down mitigating an effective anti-tumor immune response, leading to resistance to these immunotherapies. So in order to look at this, we had this very large clinical trial that consisted of patients that were either treated with anti-PD-1 immunotherapies or not treated. And within the treated group, we pathologically scored them to be either responders, meaning that greater than 90% of their tumors would shrink after their course of treatment, or non-responders. We did single-cell RNA sequencing on T cells isolated from these patients' tumors, and specifically, I pulled out the FOXP3 positive regulatory T cell compartment, which we had about 74,000 T cells in this data set. The first question that I wanted to ask was, are all of these infiltrating Tregs the same, or are there unique genetic programs that could lead to subclustering and distinct characterization of these Treg groups? And what I found was that, indeed, there was distinct differences in these tumor-infiltrating Tregs, and they range from a very activated suppressive phenotype, shown kind of in the bottom right-hand portion of this UMAP, that had genes such as GITR, which is the same as TNFRSF18, OX40, which is the same as TNFRSF4, CCR8, LAG3, very suppressive genes. There was a resting or more lymph node homing clusters on the top left portion of the UMAP, marked by genes like the CYP1 receptors, CCR7, TCF7. There was a very small type 1 interferon responsive genes, with interferon responsive genes being upregulated in these clusters. And then this other smaller Th1-like or cytotoxic cluster, which was marked by expression of genes like chemokine receptors, granzymes, and interferon gamma. From this, we then subclustered this even further by looking at the stratification by response status. Again, these patients were either treated or non-treated, and then within the treatment group, they were categorized as either being responders or non-responders. And when we looked at the relative cell densities, we found that responding tumors had a relative absence of this particular activated Treg cluster. And it wasn't all of the activated Treg clusters that was different. It was actually this activated 1 cluster, which was marked by high expression of OX40 and GITR. And when we looked further into this, we see that although many of the activated Treg clusters shared genes like CCR7, 4-1BB, ICOS, you can see that's shared between these activated groups. There really were 3 main genes that were uniquely expressed by this activated 1 cluster compared to all of the others, and this was OX40, GITR, and LAG3. NTPD1 or CD39 is also much more expressed in this cluster, but you can see its expression is shared between activated 1 as well as activated 3. Because of the unique phenotype of this particular cluster, namely by these genes, OX40, GITR, and LAG3, we developed a gene score that was anchored on the expression of these genes to look not just on the cell numbers and cell densities, but actually just based off of gene expression. Using this activated Treg score, we then again queried the gene expression profiles in the tumor-infiltrating Tregs, as well as Tregs isolated from the adjacent normal lung tissue. And what we found was that responding tumors had a significantly reduced activated Treg score compared to non-responding tumors or untreated tumors. And most notably, that this phenotype was unique to the tumor-infiltrating population, that the adjacent normal lung Tregs did not show any differences in these gene expressions. In order to look into this further, we hypothesized, I'm just going to step back for a second, that this gene expression was due to TCR stimulation within the tumor microenvironment, leading to the activation of these Tregs, which was marked by expression of OX40 and GITR, which is well known to be genes that are turned on in Tregs as well as other conventional CD4s or CD8s upon TCR stimulation. So we thought these Tregs were recognizing antigen within the tumor microenvironment, turning on these genes, and resulting in this very highly suppressive phenotype, which I've proven to be very highly suppressive in vitro, which was then leading to overall enhanced immune suppression, which was resulting in the lack of response. And if these Tregs were absent from the tumor microenvironment and not turning on these genes, then they had a more effective anti-tumor immune response, which was leading to the responding phenotype, which we are seeing here. In order to study this further, I developed a mouse model, which I don't have time to discuss the details in today, but we're using a transgenic TCR that recognized the LCMV glycoprotein. I then forced thymic expression of this glycoprotein to allow for positive selection of the Tregs, and then could adaptively transfer these marked Tregs into mice that were bearing either MC38 tumors expressing that glycoprotein or MC38 tumors that did not express that glycoprotein, to really hone in on the question of what happens when a Treg sees antigen within the tumor microenvironment. The first thing that I wanted to check was to make sure that this mouse system that I've developed was actually able to recapitulate some of the data that we are seeing in the human data. And what we found was that indeed it was. We saw this activated population, again, marked by expression of OX40, GITR, LAG3, as well as EBI3, this resting population, this interferon-induced population, and the Th1-like cytotoxic population as well. So in looking at this further, I did single-cell RNA sequencing on all of the Tregs, both endogenous as well as adoptive transferred, isolated from, again, tumors that express this particular glycoprotein or tumors that did not. And we wanted to ask the question, what genes are turned on in the specific transgenic TCR model when they see antigen within the tumor microenvironment? And what we found was actually not at all what we expected. Again, I thought that TCR stimulation in the tumor would lead to expression of this highly suppressive phenotype, but actually, all of the transgenic TCR Tregs, which are marked by these red dots, acquired this Th1-like cytotoxic phenotype. And if they did not see antigen within the tumor microenvironment, they almost all resulted in this resting phenotype and stayed more homed to the spleen or lymphoid tissues and did not actually traffic into the tumor. We looked at this further by doing an adoptive transfer model where I isolated the Tregs from the tumors either one day after they've been in the tumor, 7 days, or 14 days, and looked at trends of changes in gene expression. And what we found was that over time, these Tregs that were recognizing tumor antigens lost FOXP3 expression and gained genes that were associated with highly proliferation, so Ki67, STIM1, TOP2A, other genes like checkpoint molecules, CTLA4 and PD1, LAG3, and then this very seemingly pro-inflammatory, so almost cytotoxic in phenotype, like with NKG7, interferon gamma, granzymes, as well as these chemokine receptors. And they were losing these resting Treg genes as well as FOXP3 and CD25 or IL-2 receptor alpha. Using the gene score that we were able to identify from this mouse model, I went back to the human data to ask the question, where in the human data are these Tregs if they exist at all? And what we found was that there was this very, very small population within this Th1-like cytotoxic cluster, which we defined as SC0. You can see that represented here in the UMAP. And when we looked at the genes that were highest represented in this particular cluster in the human data, they again were very, very similar to what we were seeing in the mouse data, with expression of granzymes, these chemokine receptors, NKG7, perforin, as well as interferon gamma. You can see this represented in the heat map as well as in the red overlay here. And when you look specifically, we saw that this particular cluster, compared to all of the other Tregs, had loss of FOXP3 expression, loss of CD25 expression, and gained expression of TBET. I often get questions, and since this is a recording, I want to make sure to address this, of how do you know that these Tregs are really Tregs and not just T conventional cells that might have turned on FOXP3 transiently? And, that's a little bit hard to really address. In the mouse system, I did a lot of methylation assays, and they look exactly like Tregs on the FOXP3 promoter, as well as any of the other conventional Treg promoters like Icaros, CTLA4, so on and so forth. The other ways that we looked at this is I adaptively transferred a large number of T conventional cells that recognize tumor antigen to see what genes they would turn on. And although they did turn on proliferation genes as well as some cytotoxic genes, this particular genetic profile was unique to a Treg phenotype, not a T conventional CD4 phenotype. The other way that I looked into this in the human data was look at TCR sharing between this particular cluster, SC0, as well as all the other Tregs, as well as non-Treg CD4s. And we found that the SC0 TCR repertoire overlapped significantly with other Tregs and did not overlap at all with CD4s, suggesting that a Treg is actually becoming this pro-inflammatory phenotype, that it truly is not just a CD4 conventional cell that happens to be clustering in the Treg populations. And you're welcome to look into this further. All of those other experiments are published in this publication of reference down below. When we stratified this against by response, we found that the responding tumors actually had a marked increase of this particular Th1-like cytotoxic cluster. And although we didn't quite reach significance, I hope you can appreciate that there's a trend towards it, indicating that this particular population was much more increased in the responding tumors compared to the non-responding tumors. So to summarize what we found, one, there are many distinct subpopulations in these tumor-infiltrating Tregs that have just differing functionalities, and importantly, they have cell surface markers that could be targeted for future therapeutics. We found that the non-responding tumors have a relative enrichment for these activated Tregs that have co-expression of OX40, GITR, as well as LAG3. And overall, based off of both cell density as well as gene expression, this population was markedly reduced in the responding tumors. We found that these tumor antigen reactive Tregs using this mouse model that I developed actually acquire a Th1-like or cytolytic program, and that this population was relatively enriched in the responding tumors. Overall, we described two very distinct and opposing tumor Treg populations that seem to have very differing functions within the tumor microenvironment, one of which we have associated mechanism of tumor antigen specificity in these Treg populations, which could lead to this shift in their phenotype away from a suppressive phenotype and towards a much more pro-inflammatory phenotype. And ideally, what we would like to do is have these populations, as well as others, be used for precision immunotherapy and have novel targets to enhance the response to immunotherapy by shifting these Treg phenotypes from being very suppressive towards more pro-inflammatory. For my future research in dermatology, what I'm really excited about is this idea of this Treg plasticity, as well as permanence. And so what I found is that there are really, many different unique populations of these Tregs within one tumor, and they seem to be shifting in their phenotype due to various stimuli, whether that's TCR stimulation or co-stimulation on other molecules. Much more work needs to be further explored. But I think that what I would like to do in the future is to look into this plasticity of these phenotypes and be able to harness some of what we've learned here to understand how we could shift a Treg from being very inflammatory or shift it back towards being very, very suppressive and maintain that suppressive ability rather than losing it and becoming this more pro-inflammatory Treg. And for dermatology, I think this could be applied for a number of different things. Of course, with my background in cancer tumor immunology, melanoma treatments, especially with the advent of anti-PD-1 and LAG-3 combination therapies, I think will directly apply to this Treg plasticity that I've been interested in. But in addition to that, I think that this could be applied for many other aspects of dermatology, like understanding how we could maintain the suppressive capacity and help to treat autoimmune skin disease. Also, how we can understand how these Tregs are having tolerance to skin microbiota, involved in hair follicle regeneration, as well as wound healing. And again, how we can harness some of these things that I've learned and hope to learn in the future towards developing much more precision therapies, targeting some of these cell surface markers, or even to develop durable, permanent cell therapies in which we could have a very, very suppressive CAR Treg be transferred into the human body and be sure that they'll maintain this phenotype and not transition into a much more pro-inflammatory, less effective suppressive cell. And with that, I'll stop, and I want to thank everybody who's made all of this work possible.